Okay, thank you for coming, everybody. This is Ben Dichter, and I will give a quick introduction about him now. Dr. Benjamin Dichter is the founder of Catalyst Neuro, a consulting and software development company specializing in neurophysiology data standardization and analysis. His work focuses on developing and implementing the Neurodata Without Borders data standard, or NWB, and the Distributed Archives for Neurophysiology Data Archive, or DANDI, enabling efficient data sharing and collaboration in neuroscience. Through Catalyst Neuro, Ben leads initiatives to create and maintain open source software tools that facilitate neurophysiology research and data analysis. Today, he'll be focusing on standardization through Neurodata Without Borders and how centralized repositories like DANDI are transforming collaborative research. He'll be demonstrating how these tools can be integrated into research workflows to enhance reproducibility and accelerate scientific discovery. He'll be giving a few examples and success stories for us today. Welcome, Ben.
Hi, everyone. I'm Ben Dichter, the founder of Catalyst Neuro, and I'll be talking to you about open neurophysiology with NWB and DANDI. Before I start, I just want to mention that there's a link here, bit.ly/NWB_DANDI, which takes you to these slides. So you can use that as a reference. There are also going to be a lot of links on these slides, so I encourage you to go to this website and follow along, and then click on those links to lead you to the resources I'll be talking about. So first, let's talk about why there's a need for open data in neuroscience. So first of all, the data that we collect is very expensive. It is expensive in a few different ways. First of all, it takes a lot of money, right? It can take tens or even hundreds of thousands of dollars for some of these data sets, sometimes even millions of dollars to collect these really intricate data sets. Two is it takes a lot of time. It can take years of time, and it can also take an individual's time. You know, people who are incredibly highly trained and skilled, it can take years of time to collect these data sets. And then number three is we often use animal lives when we collect this data. So it's really our responsibility to make sure that we derive as much scientific insight as we possibly can from these data sets. And we know that many of the experiments that we run can be used to answer many different scientific questions, not just the ones that motivated the experiments. But sharing data with colleagues can be really difficult. It can be difficult to share data even within a lab. And when you try to share data with collaborators, it can be even more difficult. This can be because there's an enormous diversity of the different data formats. The data can be very large and just unwieldy to share, or the metadata could be incorrect or incomplete. Essential metadata that is kind of unimplied or assumed within a lab might not be understood by others. And so it can often take, honestly, months of back and forth communication if one lab wants to use another lab's data. On top of this, it can be difficult to share scientific software. The software that we build to process the data, analyze it, visualize it, this often relies on data being in a certain format. So you often have to convert from one format to another just to use this software, and it can be really difficult to do that correctly. Without the necessary tools and infrastructure for sharing data and software, it's actually often easier for labs to collect their own data and write their own software. And we think this is incredibly wasteful, and it slows down science in an enormous way. It hinders the reproducibility of science, and the quality of the scientific work. It prohibits us from actually building effectively on top of one another to progress science effectively. In my opinion, it's one of the major impediments to scientific progress. Um, I'm envisioning a future, and my colleagues are envisioning a future where using open data is the norm. Where if you have a scientific question, instead of thinking, what experiment can I run and what data can I collect, you think, what query can I run? What search can I run on an archive and see if there's data available already that can help me answer this scientific question? A future where if you're writing a grant, the expectation would be that you would run a search on open data sets and discuss what open data sets are available. And if you do need to collect new data, that you should motivate that in the context of what data is already available. That's not really the norm right now, but we're working on changing that. And I'm going to discuss the tools that we've been building to do this. On top of this, there's a new data sharing policy from the NIH. This is fairly recent. It started in January of 2023, and it essentially says if you are funded by an NIH grant, then you have to share the data that you collected under that grant. If you publish a paper, then at the time of publishing that paper, you also need to share the data. If you don't end up publishing a paper, then when the grant finishes, that's when you need to share the data. So either way, you need to share the data that was funded through these NIH grants. And in addition to that, when you submit an NIH grant, you need to submit a data management and sharing plan, which details how you're going to do this. Now, this is a really new policy, and it's a pretty big change from the requirements previously. So a lot of labs are trying to figure out how best to adhere to these new data sharing policies. And this is one of the things that my company, Catalyst Neuro, specializes in. So I'm the founder and CEO of Catalyst Neuro. We're a team of international research software engineers that has expertise in both neurophysiology and in software engineering. And our specialization is in open neurophysiology software and data. And we really focus on connecting the data producers, tool builders, and data users in neurophysiology research. So when I talk about neurophysiology data, I'm talking about, broadly speaking, information recorded from the functional nervous system, particularly the brain. Um, so two of the most common modalities in neurophysiology research are calcium imaging or other types of optical imaging, where you somehow genetically manipulate the cells to fluoresce when they are firing or when there are certain other characteristics. And then you can use a microscope to view the activity or the functional patterns of that neural tissue over time. Another really common type is electrophysiology, in particular extracellular electrophysiology, where you stick probes that have tiny electrodes into the brain of an alive animal. And then you can measure the voltage from those electrodes, and you can identify little blips in that signal, which correspond to action potentials from nearby neurons. So in addition to these and a few other different types of data modalities within neurophysiology, there's often some kind of stimuli, some kind of visual stimuli or auditory stimuli or some kind of task that the animal has to do, or even some kind of electrical or optical stimulation. And then increasingly, we're looking at behavior, both like task-based kind of behavior and also natural behavior. So this is used to study basically two types of paths. One is to study the brain as it normally functions, right? So to study functions like sensory processing and behavior and cognition. And it's also used to study models of different diseases, such as Alzheimer's and Parkinson's, and essentially every neurological disease, there's an animal model for it somewhere. And this is also used to study humans as well, though I would say most of the data that we work with is animal data, non-human animal data. When we look at this neurophysiology data, we're looking at both the raw and the processed form of it. So, for instance, for electrophysiology, there's a new type of probe called neuropixel probes, which are really popular because they have hundreds of electrodes that allow you to record from many different brain regions at the same time. And so when you stick these in and you record from them, you get just voltage over time from each of those electrodes. And then you need to do a processing step called spike sorting, generally, where you identify the times of the action potentials that are observed from this voltage. And then you can also separate out different neurons based on the shapes of those action potentials. There's also calcium imaging, where you're recording just basically a video of the microscope over time. And then what you want to do is segment this into cells and then identify the fluorescence trace of each of those cells over time. And then behavior, which is becoming increasingly common. We have videos of animals. It's so cheap and easy and really powerful. So it's becoming increasingly prevalent to just stick a video in there and record the animal. And now we have these incredible machine learning tools that help us track the pose of animals over time and also attributes like eye tracking and stuff like that. So it's really great to be able to integrate this into the studies. So essentially, when we're talking about neural data, we're talking about the raw data as it comes off the acquisition system. And then generally, we're also talking about some kind of process form of the data that's a little bit closer to the actual neural activity that we're interested in understanding. So there are several challenges in standardizing and sharing this neurophysiology data. The three main ones are, number one, there's a huge diversity in the data formats. Uh, we have worked with over 50 different data formats. Essentially, in neurophysiology, we're in this unfortunate situation where nearly every company that creates a new acquisition system invents their own data format of how they want to store the data into on disk. So what we have is this enormous proliferation of data formats, some of which are really difficult to read. They require certain operating systems, or they're not very performant. So that's a really big challenge when you want to share data with colleagues. They might have never seen this data format before. The other challenge is there's just an enormous amount of data. The two types of studies that I mentioned on the last slide, NeuroPixel and wide field calcium imaging, they can commonly record a single session worth of data can be over 100 gigabytes. So if you're talking about many sessions from many animals, you can easily get to a terabyte or multi-terabyte scale data set. And that's just logistically difficult to share with people. And then the third reason is that these experiment protocols are actually quite complex, and they often involve synchronizing different systems together. You often need to have multiple acquisition systems recording simultaneously, and then kind of message pass somehow between these two to synchronize them post hoc. And these are really custom schemes that it's really difficult to automate and generalize across labs. So generally, you do need some kind of custom solution to standardize basically every different lab's data. So there are two different initiatives that we're involved in. We're part of these larger teams um, that is trying to standardize and share this data. It's to build infrastructure to allow scientists to collaborate. So the first is Neurodata Without Borders. This is an initiative led by Oliver Rubel at the Lawrence Berkeley National Lab. And it's a standard for neurophysiology and behavioral data that is meant to kind of bridge all of these diverse data formats and handle the scale of the terabyte scale data sets, as well as have machinery that can handle the synchronization between these different streams that are recorded simultaneously. And then there's the DANDI archive, which is an initiative led by Satra Ghosh from MIT and Yara Kalchenko from Dartmouth. We're also on this team, and it's an initiative that is building an archive and collaboration space for sharing and analyzing neurophysiology data. So the way I kind of think about these is NWB, I kind of think of as like an MP4 for neurophysiology data. It's a standardized data format, though NWB can extend a bit beyond one data format, but it's a way of describing the data that enables someone else to just pick it up and use it, right? If someone gives you an MP4 file, chances are you're going to have software that knows how to read that and play the movie, no problem. There was this nice standardization around a few different video formats, MP4 being the most popular, and now we can easily share videos with each other. It wasn't like that 20 years ago. Now it's much simpler. And then I think of DANDI as kind of like YouTube, where any neuroscientist can submit data for free up into the archive, and anybody else can go and search this archive, find data sets that are relevant for them, and download them for free. Okay, so let's talk a bit about NWB. So the goal of NWB here is to package all data with the metadata necessary for reanalysis. So this, of course, includes the neural activity itself, right? The voltage traces from the extracellular or intracellular electrophysiology, or the images from an optical physiology experiment. But it also includes a lot of other metadata, information about the data acquisition system, about the experiment design, what stimuli, what was the trial structure, information about the subject, such as their species and genotype, age, and weight, and information about the behavior. Was the animal moving through the space? What was their task accuracy? What kind of video or audio was recorded during the experiment? And then finally, there's this ability in NWB to build extensions because neurophysiology is an incredibly dynamic field where new types of data acquisition and new types of data analysis are invented all the time. And if we just tried to develop a standard just for what was available at the time of developing the standard, it would soon become outdated. So these extensions allow you to build upon the standard and take an existing neurodata type specification and extend it and enhance it to satisfy the needs of new acquisition systems and new data processing techniques. So to give you a sense of what an NWB file actually looks like, essentially you'll have all of the data for a given session in one file that itself has a hierarchical structure. So here I'll show you inside the general group, there's information about subjects such as the species, there's information about the experimenters who did the study, and then let's say we're doing an electrophysiology experiment with a lot of electrodes implanted in the brain. You'd also store metadata about each of those electrodes. Where are they in the brain, for instance? And then once you have this electrodes table, you can build an electrical series which references that electrodes table and stores the actual voltage recordings of your experiment. It also has metadata that you can use to understand these voltage recordings, such as the unit of measurement and the conversion factor to get to that unit of measurement and the sampling rate if it's regularly sampled, or timestamps if it's irregularly sampled. And then finally, you'll have the derived data. So in this case, you'd have a units table, which stores the spike times that you extract from these voltage recordings. So you'd have the spike times, and you could also have metadata about each of these units, like the shape of the waveform and what electrodes it came from. So that's an example of a few neurodata types. NWB defines a lot of different neurodata types that spans different types of modalities, including extracellular and intracellular electrophysiology, optical physiology, behavior, stimuli, and experiment metadata. So I'm part of this team that maintains the data standard. We also maintain software around that standard that allows you to easily convert data to NWB, easily and performantly read and write data, develop and share neurodata extensions with your colleagues, and things like that, view the specification itself. And then around this NWB suite, we have a large ecosystem of analysis and visualization and data management tools that have been built to integrate with NWB. So all of these tools read and or write NWB data and are part of this NWB software ecosystem. And so a big part of our job is reaching out to developers of new types of software and helping them integrate with NWB. And if anyone watching is interested in integrating, please reach out to me. I'd be happy to offer some guidance there. And then once we have tools integrated, we like to keep track of these things. So we have a website, nwb-overview.readthedocs.io. And here you'll find a glossary of all of the core tools that we maintain, as well as all of the analysis and visualization tools that have been integrated with NWB. And there are dozens of tools here. So you can check this out and check out tools for the types of imaging modalities that you're interested in. Now, converting to NWB can be a challenge based on the challenges I mentioned earlier, the diversity of data formats, the size, and the kind of complexity. So we have an approach that we have different approaches for converting that can suit different needs. So here on the left, we have the NWB guide. So this is an application that's a no-code application. It's a graphical user interface that provides forms and walks you through the conversion process. Then in the middle here, we have NeuroConv, which is the back end of guide, where it does require a bit of coding in Python, but we try to automate as much as we can. And then we have PyNWB and MatNWB, which provide low-level interfaces for reading and writing NWB files. So the guide, we like to think of the NWB guide as kind of like a TurboTax for converting your data to NWB. So it's an application you can install on your computer and double-click to open. You don't have to go into the command line or anything like that. It's a simple interface. The first thing you will do is search across the supported data formats. We have over 40 data formats currently supported. You find the ones that are relevant to your experiment. And then we extract as much metadata as we can from those source files, and we provide you a series of forms for you to fill in any missing data, metadata that may be required by NWB or DANDI. Commonly, DANDI and NWB require metadata about the subject. What was the species? What was the sex? What was the age of the subject? Files. Usually people have to manually enter. And then we provide an interface to interactively view and explore these files to make sure that they were converted correctly, and then you can batch convert them. And finally, we provide an interface for you to create a new data set on Dandy, which we call Dandy sets, or to choose an existing Dandy set that you own and upload this data to Dandy, all within this app. And then you're done. You've submitted your data to the Dandy archive. So if you need a bit more flexibility, there's NeuroConv, which now supports over 44 different data formats. The idea is you take data from any one of these many formats, and just with a few lines of code, here we're telling NeuroConv where the source files are. We're extracting metadata from those source files, and then updating or editing or adding some metadata, passing that back into NeuroConv, and just telling it to run the conversion. So just in a few lines of code, you can then produce an NWB file that uses lossless compression to minimize the disk space used without changing the actual data values, and it optimizes these files for cloud compute. And then finally, we have the low-level interfaces, PyNWB and MatNWB, really useful for custom data such as trial structures, responses, lab-specific behavioral metadata. This is what you'd want to use if you want to build an extension to NWB to handle new types of data acquisition or data processing. We have lots of documentation and tutorials for each of these to handle all sorts of different use cases. Okay. Now moving on to DANDI. It is the Brain Initiative archive for publishing and sharing neurophysiology data, including electrophysiology, optophysiology, behavioral time series, images, and immunizing experiments. So contributing to the DANDI archive. So DANDI requires neurophysiology data to be neurophysiology data and associated behavioral data to be in the NWB format. It validates these files. It checks them for best practices, and it also extracts metadata from the NWB files because, of course, they're in the standardized format, and that allows DANDI to provide sophisticated search over these files using the metadata that can be extracted from NWB. So in DANDI, a neuroscientist can upload hundreds of terabytes of data to DANDI for free, which I know sounds maybe too good to be true, but it is true. Are working with the AWS Open Data Sponsorship Program. Essentially, this is a program that Amazon runs where they work with scientists to build archives for open scientific data, and we can upload and download to S3 buckets for free. And so we pass that on to the community. We want to make sure that this is useful scientific data, but if it adheres to our quality and formatting standards, you can use DANDI for free. And and this is really great because it's enough space to store all of the processed and raw data for most of the current experiments in neurophysiology. So this really is an enabling resource that allows us to go beyond thinking like, well, how can I share, how can I package this data so that it's not too big, but it has essential information. You can just really share all of it, which is great because it allows someone else to then go start from raw data if they want to and redo spike sorting using their own techniques or redo image segmentation using their own techniques. So you can upload data to DANDI using the NWB guide, and there are also programmatic interfaces if you want to integrate it into your own software or do something more automated. Now, we've seen a lot of adoption of NWB and DANDI here. Here I'm showing on the left, this graph is just the number of data sets on the DANDI archive. So in black here, you can see we have this nice superlinear curve. We just recently crossed 300 data sets on the DANDI archive. And then blue, using the same y-axis, because coincidentally they're equivalent right now, is the number of terabytes on the archive. So we have about We have over 300 data sets in NWB with data publicly available on the archive, and they comprise over 300 terabytes of data. So this spans many different species. A lot of it is mouse and rat, but we also have some human data, and then monkey data, fly, and then you get into lots of other species, some of which you might recognize as model organisms such as C. elegans and zebrafish. I think at some we have like one or two of ant data. We've got all sorts of species in the archive already. These data sets, they span from generally tens of megabytes to tens of terabytes. And they span different modalities as well. The highest is extracellular electrophysiology. We also have a decent, about 70 optical physiology, lots of calcium imaging data sets. And we have a decent number of intracellular electrophysiology as well, such as patch clamp recordings. Some highlights here, some really high value data sets that are available for free on this archive. One is the International Brain Lab Brain-Wide Map, which is a collaboration between dozens of different labs to collect this really high volume data set using neuropixel electrophysiology, eye tracking, and behavioral monitoring. Essentially, they have these mice doing a visual decision-making task, and they are recording from all over the brain, thousands and thousands of neurons. We have over 33 terabytes of data from the International Brain Lab that we're hosting on DANDI. The microns data set is really interesting. So this is an experiment where they showed images and videos to a mouse and did standard calcium imaging, and then they took that neural tissue and they did electron microscopy to have a detailed structural view of that same neural tissue. So this is a really revolutionary data set because it allows you to make associations between the structural connectivity and the functional connectivity on the single neuron scale. And the functional data for this data set is stored on DANDI. And then there's this really great program that the Allen Institute is doing called OpenScope. And they have actually more than this, they now have 7 different data sets on the DANDI archive. So the OpenScope program is really unique. It's this program where scientists can propose experiments to OpenScope, which is run by the Allen Institute, and they choose the ones that they want to do the most, and they will use the Allen Institute's enormous, sophisticated experiment infrastructure to collect these data sets in a really high quality and standardized way. They generally use neuropixels or 2-photon calcium imaging. So the people who propose this experiment, they get exclusive access to that data for some amount of time. I think it's a year, and then after that, the data set gets open to the community, and the OpenScope program shares this data with the community using NWB files on the DANDI archive. And so there are currently 7, and I'm just going to actually click this to show you what the DANDI archive actually looks like. And yeah, so here are the 7 that are currently available. You can click in to get additional information about each one of these, a detailed description, contributors, funding information, associated resources, and then metadata that was extracted automatically from the NWB files. And here we have, this is a 2.5 terabyte data set. So really large data sets with lots of great information inside them. And yeah, it's really easy to search this archive. I mean, just go to dandiarchive.org, and you can just say like hippocampus, and then boom, you get dozens of data sets that are involving the hippocampus. I mean, it's really that simple. You can get a more fine grained to find things that you're really interested in. Okay, in addition to this, the OpenScope has this resource called the Data Book, which is a book, a Jupyter book that demonstrates how to read their data, how to do basic data visualizations, basic first order analyses, and it even goes into much more complex analyses. This is a really great resource if you're getting started with converting neurophysiology data, because you get tons of code to start out with. So just for instance, this chapter, I'll show you to access optical physiology data. and do visualizations of responses to stimuli. Okay, another great resource here is called NeuroSift. This is a tool built by Jeremy Maglin, who works at the Flatiron Institute. So essentially what this is it builds interactive, intelligent visualizations of NWB data. So it can understand the structure of an NWB file. It knows about the metadata associated with different types of data, and it can render reasonable visualizations for any type of data that you throw at it. It runs in the browser. There's no need to install anything or run any external services. You can literally just pass someone a link and they can view it. It streams data on the fly directly from the DANDI archive, so there's no need to like download really large data sets. You can view small portions of large data sets without having to download the entire file. And it's integrated with the DANDI archive website. So I just want to show you two demos of how NeuroSIF can help you explore some of this open data. So the first demo I'm going to show is this data set, which is a data set from one of the papers that was cited in the Nobel Prize to the Moser Lab. So this is where this is one of the first papers where they demonstrated grid cell representation in the entorhinal cortex. Essentially, they have a rat walking around a large cage with a position being tracked. They also have electrodes implanted in the entorhinal cortex, which is recording electrophysiology data from that brain region. And they have action potentials extracted from that signal. So I'm going to go to the Dandy set landing page here. And what you can do is you go to Files, and then I'm just going to pick a random subject and session here. I'm feeling lucky with this one. All right, NeuroSift, open with NeuroSift. Okay, great. So we have the acquisition electrical series, and then I'm going to go into units, and I'm going to show a raster plot of the cells that were extracted from that electrical series. And then for behavioral data, I'm going to just click both of these, view items. Now this is streaming data directly from the archive and rendering these visualizations. So I'm going to zoom in a little bit so we can render a bit faster. These ticks here are four different neurons, the timing of their action potentials. This is the raw electrical signal. Here is X and Y position over time, and then here is X versus Y position. So you can click on here and look at the timing. Of course, these are synchronized with each other. You can press play to view this. And this really helps you get a handle on the types of data that are in these files and what you can potentially do with them. Now, of course, your analysis is not going to end here. This is really just more the exploratory phase. Let's say you actually want to get a hold of this data. So I'm going to go and just click raster plot. So this just renders this one visualization. But now you get this button load in Python, and you can literally copy and paste this code, and it will stream in all of the spike sorted data for you into Python, ready to go. So and then you can do whatever analysis you want from there. So it's really quite simple to get started with this. Okay. The other demo I want to show you, which is I think really great, is a peristimulus time histogram on the International Brain Lab brain-wide map data. So here we have mice in a task where they are shown images of varying contrast on the left and right. And they have to say whether the image was shown on the left or the right by moving their wheel. Now, it's really easy to do when there's a high contrast image, and it's very difficult to do when it's a low contrast image. And they, of course, get a reward if they're correct in their pixel brain recording electrical signals from many different neurons at once. So I'll just take a look at this. There's again over 33 terabytes of data here. We'll go to a random session. All right, open with NeuroSift. Now, there's a really cool visualization here called a PSTH, which allows us to look at the kind of the representational properties of the different recorded neurons. Takes a minute to ramp up. align to the stim on time and the go queue time. And then I'm going to group these by contrast left. You know what? Let's start with grouping by choice. And then 0 is like an error trial. So let's move that up. Okay, so here you can start to see all of the different neural activities. So this is unit 1, aligned to the stim onset time and aligned to the go queue time. And you can see there's this really beautiful reduction of activity following the stim on time. So this up here shows each individual trial, and this down here shows an average across those trials. And we'll probably find neurons that respond differently for left versus right. Do, do, do. So this is interesting. It responded differently, but before the stim time, um, which is fascinating. And then you can group by other things. So I'm going to group by contrast left. Maybe we have more neurons that are kind of selective to different contrasts. Let me find a really good one. No, these aren't as strongly tuned as I usually see, though I do see something here. Um, I don't know. That's interesting. So here we get this nice locked response to the stimulus onset. And we do see different responses potentially for different types of contrast. So anyway, this allows you to explore the data and fine-tuning properties of the units within. So this is fascinating. You get a larger response potentially for a lower contrast stimulus. Okay, anyway, I could go on. I could explore this for hours, but I will move on. I'll let you do that. Okay. In addition to this, the DANDI also offers this thing called the DANDI Hub, which allows you to, it's a free cloud compute resource at hub.dandiarchive.org. You do need to register for DANDI to use it, but it's free. If you go through the registration process, you'll have access to the DANDI Hub. You can analyze data sets in Python, MATLAB, Julia, R, or really many other different programming languages. It has particularly fast access to stream and download data from the DANDI archive. It has different server options, including a GPU server option. Please don't use it to mine Bitcoin or train the next DeepSeek. Just use it for data analysis, please. And it allows you to create notebooks that demonstrate analyses where you can read in data and do your own custom analysis and then share those with the community. So now we have now 25 different DANDI sets that include notebooks. So we have this website that indexes these. So if you're interested in any one of these, let's see. Um, Corticocellular loop for motor planning. We have these notebooks that you can then go into and use this as a starting point for your data analysis pipeline. So you can see here how you would read data in from the NWB files and potentially maybe reproduce some key figures from papers or just do some exploratory data analysis. So this is a great jumping off point if you're interested in using one of these data sets. So we're really interested in promoting data reuse. We've found now 30 different papers that have used open data in DANDI in NWB from the DANDI archive. Uh, we're really interested in promoting this. And one of the challenges, though, is that often when people use NWB data, they don't cite it. Like what we really like is for people to cite it as if it were a paper. Often they'll just like provide the URL, which is not terrible, but it's difficult for us to track the usage, which we're really interested in doing. So what we find is that and there are lots of different ways to reuse this data. So one is a data showcase where you can demonstrate a different analysis or visualization software on many different data sets. Two is a pilot. Let's say you want to do a study, you want to collect data, but maybe you want to test out your analysis on open data first so you can hone your ideas before designing your experiment. Three is a follow-up. Let's say there were some unanswered questions that were mentioned in the conclusion of a paper that the original authors just didn't have time to go into. You can access their data and follow that up and do really powerful science that way. One of my favorite ways of using open data is confirmative. So let's say you have a study, you have your own data, you have your own analysis, just a normal systems neuroscience paper. And then at the end, you search the archive for similar data and you try to reproduce those findings given someone else's data set. I really like this because it doesn't really require you to modify your research practices at all, except for at the very end. And and it has the ability to dramatically strengthen the persuasiveness of your paper, right? Like, what are reviewers going to say? I mean, there's certainly, it really alleviates concerns that there was some kind of nuance of your experiment that was the reason for these findings when you can reproduce them in a completely different data set that you had no involvement in. Or you could also use it contrastively. You could say, look, this finding is true here, but it's not true in this other brain area or this other species or with this other cell type. Um, you can use a contrastive analysis to do that. Or you could do an aggregate of analysis. You could combine data sets from multiple different sources to gain statistical power. And finally, one of the great ways to use this data is as an educational resource. You can build courses where the exercises are actually done on open data. And it's really inspiring, I think, for students to be able to use real data and to kind of walk in the footsteps of scientists that came before them using the very same data that those scientists were using. So we're collecting different ideas for data reuse. We have a forum here that you're welcome to check out or contribute to. And I'm also organizing this event called Neurodata Rehack. So this is the fourth installment of Neurodata Rehack. It's a hackathon for generating new insights from existing neurophysiology data through secondary analysis. It's open to many different types of researchers, including grad students, postdocs, RAs, engineers, basically anyone who does data analysis. The instructors are people from the NWB and DANDI teams, data contributors, and developers of analysis and visualization tools. It's long funding and meals are provided. We have done this at Genelia before. It's a really fantastic place to do it. It's a beautiful, the food is amazing. They're very welcoming. It's really ideal. In fact, this last event we did at Genelia was unanimously rated 5 out of 5 by all of the attendees. So people really enjoy their time there. And actually, this year, I just found out this yesterday, but the Kavli Foundation has agreed to fund Neurodata Discovery Awards. So these are $50,000 grants that are available exclusively to attendees of Neurodata Rehack and are intended to fund the continuation of projects that are initiated during this hackathon. So I encourage you to apply. Deadline is February 12th. We do have some limited funding for travel awards. We're probably going to try to use that to help people from other countries, and in particular countries that where it would be economically difficult for them to attend otherwise. Yeah, so with that, I want to thank the support of course, the Brain Initiative and the rest of the NIH, the Kavli Foundation, Simons Foundation, and Michael J. Fox Foundation, who have been really instrumental in the mass adoption of NWB and DANDI. And of course, the rest of the DANDI and NWB teams, in particular, Satra Yarek and Oliver Rubel. And there's a whole community of contributors here, including the NWB Executive Board that has been really instrumental in the strategy behind all of this. Um, and I have here a bunch of links that you can use to contact us if you need any help. We have a help desk, we have Slack. We're happy to do one-on-one consultations. We have a newsletter. We have documentation and tutorials. We have all sorts of things, upcoming events. And I have here links at the end for two different tutorials. One is for how to use the NWB guide to publish your neurophysiology data, and another is a demo for how to stream data from the archive and read it and do some basic analysis and visualization of the data. Okay, that is um, that's everything. Thank you for listening.